limited amount of DNA can be transferred from one phage, we can say one bacterial cell to the other via bacteriophages. Site-specific recombination events are, however, required in case of specialized transduction, which may allow the incorporation of DNA at specified sites. And the best example is of lambda phages. Now the next is the transposable elements. Transposons. As the history states, transposons were first discovered by Barbara McClintock in the samples of Zea mays. Transposons are also known as jumping genes, since they have the capacity to jump from one particular chromosomal material to the other. These transposable elements, they may be of two types, either simple or composite. Very simple transposable elements, they are simply insertion sequences. They are known as IS elements. And most of the transposons, they have IR or inverted repeats, which are the regions of symmetry on both the ends of the transposons. In case of composite transposons, such as the transposons of TN1, TN3, TN5 or TN7 family, they contain a specialized gene, as well as some additional genes are also present on the transposons. Transposase is an enzyme that is coded for by a specific TRP gene, and due to the presence of these transposase enzymes, the mechanism of transposition takes place, via which these very small elements can jump from one chromosomal material to the other. And this is how this method is also known as the cut paste or copy and paste method for the gene transfer. They are very useful in the sense that they are found within several yeast cells. They are found within higher eukaryotes such as Zea mays and some animals also. And this is one way of introducing the genes from one eukaryotic animal into the other, or prokaryotic organism into another prokaryotic organism, which may modify their genotype, and so does the phenotype. Here you can see, as you can see, there is one of the best examples of transposase gene that is present on the transposon. Now, integration and excision, they are also requiring specific sites on the host, via which there is integration of the entire transposon within the host DNA. Once integrated, it can be also excised in the similar way. Integration and excision may depend on the presence of specific sequences within the host as well as the transposons. Here you can see, after integration, the excision is also possible in the same way. Here you can see all the three diagrams, in which the first figure shows that the transposon, it is now ready to integrate within the host chromosomal DNA. Second, you can see the site-specific recombination allows the transposons to enter within the host. And third, excision can also take place in the same manner. Microinjection method. Microinjection method, it is the introduction of we can say a single gene or a combination of more than one genes from one host or one member of the same species, or we can say from the different species, into the pronucleus of a fertilized ovum. However, the insertion of this genetic material or DNA is we can say a random process. The probability of this gene that is introduced will depend and will not insert itself into a site on the host genetic material, and that will permit its expression. However, we can say that the manipulated fertilized ovum is transferred into the oviduct of a recipient female, or we can say the foster mother. Here you can see the figure in which the microinjection of DNA is taking place, and the egg is immobilized by, we can say, suction applied through a pipette. And after the suction is applied through pipette, we can say you can see the tip from where the microinjection is taking place. Now, recombinant DNA technology. We all know that today there are a lot of things available commercially, and that are mostly recombinant in nature. For example, for say insulin. Recombinant DNA or rDNA molecules, they are the DNA sequences that result from the use of laboratory methods. We can also call these methods as molecular cloning, to bring together the genetic material from multiple sources or various we can say sources. Here the creating we can say production of sequences are we can say done on the basis of homology between the host. For example, sometimes the prokaryotic genes, they mostly are containing only the coding sequences, and in which only the required genes are there, whereas in eukaryotes, coding and non-coding, both the sequences are present in the form of introns and exons. So further processing is however required, in which the non-coding sequences have to be removed via splicing mechanisms, and then the construction of vectors can be done. Clones, they have or we can say recombinant DNA molecules, they differ only in the sequence of nucleotides within that identical overall DNA structure. Here there is one example in which the vector DNA is there and the gene of interest is there on the right side. Now for processing purpose, both of them should have homologous sequences that may allow them to combine together to produce a circularized double-stranded DNA molecule. And the introduction of these recombinant vectors into the host may require one of the various techniques. The techniques sometimes may require electroporation methods or treatment with calcium chloride that may increase the porosity of the cell towards the recombinant DNA molecules. Once the DNA molecules are taken inside, the constructed vector should also have some limited size, and on the basis of that it can be expressed within that particular suitable host. So vector and the gene of interest and we can say the host, they are decided on the basis of the type of gene and its expression, and the suitability is also found out. Now tools for our DNA technology, and the first topic is cutting of DNA or processing of DNA. Here rDNA technology uses one of the variety of techniques for cutting the DNA, especially in case of recombinant DNA technology or gene cloning. The DNA is precisely cut via using molecular scissors in the form of restriction endonucleases. Restriction endonucleases affect the DNA at specified sites. Majority of restriction endonucleases have the ability to identify the DNA in the form of hexanucleotide or tetranucleotide sequences, and they can produce specified cuts. As you can see in the figure, the DNA can be cut to produce either staggered cuts or blunt cuts. Now these cuts may allow these cuts can be introduced within the vector as well as the gene of interest, so that there are ample chances that both of them can recircularize and produce a cloned vector or vector containing the insert. Now second is, as you can see in the figure, then comes the joining. After cutting comes the joining. Joining may require specific enzymes known as glue enzymes or glue-like enzymes, which mostly DNA ligases. Ligases, they introduce the DNTPs or deoxyribonucleoside triphosphates, as well as they use the energy in the form of ATP or NADH, and this is how they carry out the DNA molecule complete, and there are no cuts or nicks within the DNA if they are joined via using some glue-like enzymes such as DNA ligases. Here in the next figure, as you can see, on the right side there is an insert that is also processed by the same enzyme, via which the vector is also treated by the same enzyme, and the insert is also treated by the same enzyme or restriction endonuclease. After processing, they are allowed to religate at a specified temperature, at which there are chances that, depending on the homology, since they will be homologous, they will bind and produce a recombinant DNA. Recombinant DNA technology, tools and techniques. Once the vector with the insert is created, it should be introduced within the host. Now the vectors are of various types. The vectors may be simply we can say plasmids such as PBr322, and it may be cosmids, phasmids, or YSE yeast shuttle vectors are also there. So variety of vectors are available. and the vectors are chosen on the basis of suitability of the experiment. Now, as you can see, the primary task of constructing vector with the insert has been now over. Now, second is the preparation of choice of host organism and cloning vector is done. Second is preparation of vector DNA. Third is preparation of DNA to be cloned. These are the topics that we have seen just now. Then comes the introduction of recombinant DNA into the host organism. Now, the vector should have some selectable markers also. These selectable markers may allow the identification of cloned cells versus non-cloned cells. So this is how we can identify the cloned cells by using a specified media that may contain, suppose for say, if the vector contains two selectable markers such as tetracycline resistance gene and ampicillin resistance gene. And when we introduce the gene of interest within the vector, the clone is having only one selectable marker, the other one is disrupted at the time of introduction of the foreign gene. So on the basis of disruption of one vector, or disruption of one of the gene, we can identify the clones on some selective media that may contain ampicillin or tetracycline. Now next. Now you can see in this figure that the foreign DNA is also processed by the same enzyme EcoRI, and the plasmid is also processed by the same enzyme. Now after introducing a cut in one of the selectable markers, the insert DNA is the gene of interest is now inserted into the plasmid. The plasmid now recircularizes. Now this plasmid containing the insert is now introduced within the host, suitable host. Now there are chances that out of several hundred cells, only a few of them will be containing the insert. So they can be identified by using some selective media that contains an antibiotic, and this is how you can select a particular organism or host cell that is genetically engineered, or we can say that is a recombinant one or a cloned one, and we can amplify those kind of cells using various techniques. However, plasmid DNA and host DNA both can be expressed in the same medium. However, there are specialized we can say antibiotics such as chloramphenicol, which will amplify the plasmid genomes versus the host genome. So there are various techniques for selection of cloned cells versus non-cloned cells. Now I hand over the topic to Professor Jimish Pathak. He will now discuss the applications of genetically modified organisms, plants, and all the other remaining applications. Thank you.
Thank you very much, sir, to hand over it to the topic. Now, as my colleagues have discussed in very detail about the various techniques which we have used, or which is generally used to modify these microorganisms. So now what has been done in the past or by using these techniques, or what can be done in the future, or what might be its effects or the results that we see in the applications, the applied area in real, means what is its exact applications, or how we are practically applying this method, or what we can get the benefit regarding this. So as you can see in the list on your screen, so there are various main groups. If we'll see, then we can divide it into the generally it might be either microorganisms, plants, or animals. Then the common word we are using, organisms. So by applying all these methods, we basically one by one discuss various groups. Like first we'll start with the genetically modified microorganisms, or shortly we call it that GM microorganisms. Same way, or the genetically modified plants or genetically modified animals, or it might be genetically modified humans. So though it's not the exact modification of humans, but we'll see what is the exact meaning of that, or why we have written it is like GM humans. So first we'll start with the genetically modified microorganisms. So this microorganisms means it includes all about like the bacteria or yeasts or molds or any microorganisms. But if basically we see the meaning, means the first on which type of microorganisms it was these techniques were applied, it is mainly to the bacteria, because particularly due to their very simple or we can say their known sequences of genes. So we can say the genetically modified bacteria were the first organisms to be modified. So now if we'll see the use of such bacteria or the microorganisms, so these organisms are used for the several purposes and are particularly important in producing large amounts of pure human proteins, as my colleague have discussed earlier or giving the examples of insulins. So which is, if it is genetically produced and if it is used for the humans, then it is known as humulin. So humulin is also produced with the help of recombinant E. coli. So this is some of the list of all the products or various groups of products, so like insulin or hepatitis B vaccine or tissue plasminogen activator or human growth hormone or ice minus bacteria. It's not the product but it's the microorganism itself, and then interferons or the some of the processes we can say like the bioremediation or bioaugmentation, bioleaching, biopolymers or industrial products. So in bioremediation it is the or bioaugmentations, these are the processes in which generally maybe it is used to remove the pollutants or to for the cleanup or sustainable development of the environment with the help of genetically modified microorganisms. Bioleching is also the same process, but it is used to extract some metals from the low grade ore, which is chemically not possible for us to extract that metals because it is economically not feasible. Same way we can also produce biopolymers with the help of genetically modified microorganisms. Or industrial products means it includes all the products various like ethanol or enzymes or organic acid like citric acid. So all various products can be done or produced with the help of genetically modified microorganisms. It is possible that they might be either produced by mutation or any genetically modified techniques, as earlier we have discussed. So first, as earlier I have discussed about the humulin or the human insulin, as you can say it is produced by a recombinant E. coli cells and by altering its genes. Also now this is the, you can see the some brand product which is commercially available